Zamir Punja. It's so nice to see you again. Thank you for coming on MyFlora Fridays. This should be fun. It's been a few months since we hung out at MJ Unpacked in St. Louis. You're one of my favorite scientists to follow in this space. I have thoroughly enjoyed all of your publications and following your journey looking at pathogens, specifically in cannabis, at least as of lately. You've got an extensive list of publications, right? And I think I remember talking to you one time, and it was during COVID. You said you had nothing much to do other than sit around and write. I think you published something like 20 plus papers during COVID, during the shutdown. Is that correct?
Yeah, so I mean, it's been a good road. It's been a good run. As most people that work with cannabis will tell you, there's so much to discover. There's so much to know. There's a huge learning curve. When I started doing this in 2018, and that year is important because that's when Canada legalized cannabis or marijuana for recreational use, and that opened up, lots of opportunities for research. And I just, I want to use the word stumbled. I just sort of stumbled across a grower that was experiencing, believe it or not, Fusarium issues on his plants. And he says, I hear you're a plant pathologist, and can you come over and look at my plants? I went over there and checked it out. And from then on, it was disease after disease after disease that I was coming across. And early on, people would tell me, "Oh, you don't have to worry about cannabis or hemp. They're no pathogens." But obviously, we know that's not true because there wasn't any research, right? Nobody had looked
Mm.
underground. Nobody had looked to see what was there. So started with Fusarium, moved on to Pythium, and then of course powdery mildew. And just when I thought things were going okay, hop latent viroid came along, and so there was a continuum of pathogens, which when COVID came, it kind of shut things down a bit. I still could go and visit their farm. I would
Mm-hmm.
go with the masks and gloves and all that. So I wasn't prevented doing that, but there wasn't much else to do. So I started writing all this data that I collected. And
Yeah.
unlike a lot of researchers, I actually like to write. I actually love to write. And so I got, like you said, I don't know, 15, 20 papers out over the 3 years, that 2.5 years that COVID hit us.
Yeah. That's awesome that you like writing. I like writing, but I'm very bad at it because I have a tendency to like word vomit and overshare, and I put like
Yeah,
a lot of emphasis where maybe the emphasis isn't needed.
yeah.
And the thing that bothers me about writing is you spend, a year or two investigating and working on something just for it to end up being like one or two sentences in a paper that you publish.
Yeah, I mean, it is a bit of a skill, and I re-edit and re-edit myself a lot. So I'll write things, like you said, just a whole blurb, and then I'll come back next day and go, oh, this doesn't look right. And I'll go back and edit it, and I'll cut it and I'll cut it until it reads really well. And so
Hmm.
it's a process, it's a learning process that a lot of students don't have when they start, but then they pick it up as they go through. And to be honest, writing is really the important component of research because if you don't get the word out, how will the growers know that they've got a new fusarium or they've got some other species of powdery mildew or hop latent that's just broken out?
And the science communication portion of it, to be able to effectively communicate with somebody who's not classically and scientifically trained and doesn't have the jargon, vocabulary that so many scientists use, and even in different fields. Like if I'm reading, a virology paper, I'm like, I don't even know what they're talking about. Like,
Well, but, and this is where podcasts and things like what you're doing come in really useful because we can narrow it down. We can distill the information without using all these technical terms. And I've
mm-hmm.
been on, I've been on a lot of podcasts and webcasts since cannabis came along in my research. And so I'd never done a podcast before. I'd never done a webinar. This all came about, well, partly through COVID, but mostly through cannabis. And
Yeah.
so, I'm in there with CanMed, I'm in with you, I'm in it with other organizations that see the need for exchanging information. And, I have no problem telling people what I found. A lot of people are secretive. They go, oh, well, don't share this information. And I said, you've got to share it. If I didn't put out the hop latent viroid report after we heard, from California that this was a problem, where would we be? I mean, you need to tell people what we find. And so I haven't been of the mindset that I'm going to keep it to myself. I just want to share it.
Well, and especially in an industry like cannabis, when we're so far behind in the research because of we haven't been able to research it for, the better part of 100 years
Yeah,
since we've started, since the development of really good scientific tools to diagnose things like pathogens,
yeah,
qPCR and stuff like that. And so I feel like we need to work together to kind of catch up. And
yeah,
in a space where you're doing everything in secret, you could have multiple scientists working on the same thing and just wasting time and money when we could all be working on all these different
yeah.
bits and
But, e- but
advancing
even but even
so
then,
fast.
surprisingly, there's not a lot of researchers that have taken on cannabis. I mean, part of it is that, it's still regulated, even
Hmm.
here in Canada, right? We need permits and we need licenses and things like that. But there's only a handful of researchers in Canada and just even a handful in the US that have
Hmm.
attempted to get on. Now, of course, hemp is different. Hemp being legal, there's way more researchers on hemp starting up, less so on cannabis or what we call marijuana, which is the high THC. There's
Hmm.
less researchers working on that.
Yeah, because that's the one they're not allowed to touch.
Yeah, yeah,
And well, and at the university level, you can't the the problem is that, working with something that is federally illegal
yeah.
jeopardizes federal funding because now you're touching drugs.
Yeah, well,
Right.
I hope this change from level 1 to level 3 happens because then that will reduce it down to, the same level as Tylenol 3 or something that's not as scary as it is now. But we'll see what the Trump administration does on that. I think that would be a bonus for research, being able
Yeah.
to at least not worry about the all of the red tapes that goes along with it.
I've been asking most of my guests what they feel about the Schedule 3 change and also the changes in the hemp definition that have also been proposed. Both of those are problematic. The hemp definition that is going to exclude seeds from THC type cannabis and regulate that as marijuana, that's problematic. But Schedule 3, I've heard a mixed bag because if they move to Schedule 3, it does open up a little bit for research, but you would still have to obtain a DEA license and jump through all of those hoops, which are not easy hoops to jump through either.
Right.
So I'd love to hear your take on how you feel about using hemp as a proxy for marijuana. I mean, they're both the same species, bred for different uses. And, traditionally hemp was for industrial purposes, so fiber and seed oil, not the cannabinoid oil, but seed oil. So those are quite different, and they actually genetically separate out really well from the types that were bred for the female flowers. But if a university is looking at hemp types that are CBD type or CBG type or even cannabinoid void types, how do you think that's a good proxy, or do you think that they're actually quite different and it's not working terribly well?
Well, I mean, in general, I would say there's a significant overlap. I mean, if you're looking at obviously cannabinoid THC biosynthesis and you're studying trichomes and stuff, that would be quite different because now you're looking at a different beast altogether. But with diseases, surprisingly or unsurprisingly, they're all the same. What we find, they'll eventually find outdoors, and what they found outdoors, will eventually find indoors. The one exception might be hop latent viroid. I'm still of the opinion that people haven't done significant research on hemp outdoors to really know what the significance of that viroid is. I mean, it's out there. It's in Colorado, it's in California, it's in Washington State,
Everywhere.
but we don't
It's
know the
everywhere.
extent of damage compared to indoor cannabis. But other than that, powdery mildew, Fusarium, Pythium, tinea, we're seeing all similar diseases indoors and outdoors, which leads me to believe that, at least from a genetic resistance perspective or susceptibility, they're quite similar.
Okay. All right. So we've been really aggressively hybridizing and breeding and selecting for single traits. Mostly it's been THC and more recently looking for more interesting terpene profiles. But do you think this aggressive selection breeding, you know, path that we've been going down has led to increases in disease susceptibility in cannabis?
That's hard to know for sure because, well, let me give you the example of indoor or greenhouse cannabis. I see a lot of differences amongst genotypes or strains in their susceptibility to disease.
Hmm.
We see some stand up really well to powdery mildew. I see others stand up really well to hop latent viroid. Now, cannabis maybe has more diversity in terms of the breeding that's occurred to enhance THC and other characteristics might have left a wider spectrum of germplasm. I don't know. I don't know enough about hemp to say that they've narrowed down the selection to less genetic diversity, and that might increase disease. But in cannabis, certainly indoor cannabis, one of the reasons we see a lot of diseases is because there's so much of it. There's so many plants, and they're all uniform and they're all grown under one environment. And once a single pathogen gets in, it just continues on and kind of wipes it out. But in terms of diversity, I see quite a bit of differences in terms of pathogen behavior on different strains of cannabis.
Hmm. Yeah, I think is it was it one of your papers, the powdery mildew resistance, where you looked at several different strains and some were not as susceptible or
Yeah,
didn't get
yeah.
as infected as others?
Yeah, for sure. So if you look at the Kush types versus other types, there were definitely differences in terms of how they responded. And now we know that it's due to, at least a couple of resistance genes or resistance loci that have been discovered. And there may be more. What we
Hmm.
don't know though is this whole concept of pathogen races. That hasn't been looked at a lot in cannabis or hemp. It was still early on because obviously the fear would be if you started growing a genotype that was presumably resistant, would that encourage more races to develop, like you see in wheat and barley and other crops? So right now, the resistance to powdery mildew at least seems to be holding up, but we'll see.
So when you say races, do you mean like the evolution of the disease to
Yeah,
become
just so
more virile?
genetic variants in the pathogen. Yeah, so that
Hmm.
because we're now all of a sudden growing a new genotype or strain, we're now encouraging the development of other more virulent pathogens. I haven't seen that yet. When I look at PM, even hop latent, we know it's quite genetically uniform. Um,
But it is possible, right? And I see
yeah,
so many parallels to, everybody's super familiar with COVID now because we had to live through all of that. Indoor versus outdoor grow. You can think of, sitting in a packed restaurant, one person has COVID, everybody's gonna get it, right? If you're outside in a outdoor seated area, somebody with COVID, you're less likely that
yeah.
everybody outdoors are because you're more spread apart, there's airflow and stuff like that.
Yeah, so I agree. Despite all the horrible things that happened with COVID, it caused people to understand more viruses. They know what the term PCR is now.
Hmm.
When I talked to one of the growers and I says, "Oh, I think we have a new virus," he goes, "Oh, is that the same as COVID?" I says, "Yeah, you could look at it that way. It's going to spread on your fingers. It's going to spread from plant to plant, and that's helped a little bit to help us understand the pathogens and what they do
And how they move
and how they
from
move. Yeah,
like throughout the environment, whether that's, a spore that gets into the air or whether you have to touch it and then touch something else. And so I
yeah,
feel like people are more attuned to sterile techniques and being wary of not, spreading taking removing your gloves like in between,
yeah.
things that you're
Yeah.
doing so that you're not or you're minimizing the spread of something.
And they're aware of damage too, right? In terms of COVID, we lost a lot of people lives and long COVID and things like that. They understand that these pathogens can also kill these plants fast.
Hmm.
You get a Fusarium outbreak, and within 7 days, you've lost a lot of plants.
And I don't think, okay, so hop latent viroid is the big scary one, but I've been wondering how serious are people taking it because I've been looking at some numbers and you can lose and correct me if I'm wrong, you can lose up to 30% of your revenue or more, 30% of your THC because the disease it wrecks the trichomes, right? It deflates the trichomes and then so not only are you losing yield, 30% of your yield, you're losing 30% of your THC. And so, monetarily wise, that can really hurt your bottom line, right?
Yeah, and I've seen a lot of growers, a lot of companies go out of business, and they haven't quite said, "Oh, it's because of hop latent." But as you said, it's because the revenues went down so significantly that when they started looking at the bottom line, you're looking at a 30% reduction in your THC levels. I guess the really hard part with that disease is that it's not obvious. And good growers will notice the flowers are not they're dudded, they're not growing as well. But for
They're
the
not.
most part, the plant grows, it's still pretty tall and produces a lot of leaves. And unlike other diseases where you look at it and you go, oh, there's something wrong. So that's the
Mm-hmm.
problem with hop latent is it's not totally obvious that you've got
Right.
something until you start seeing the reduction in your revenues, for example. I hope most growers realize that there are testing labs out there, there's testing kits out there that if they have any suspicion about hop latent viroid, they can reach out and get one of these labs to test their plants to make sure that it's there or not.
Yeah, and I was looking at a client's, well, I was somebody who reached out. They had noticed that their flowers were stalling and like just kind of they weren't bulking out. They were just like they were there, but they weren't bulking out like you would expect during the flower cycle. And they called it flower, yeah, flower stalling is I think is what they called it. And I looked through the pictures and they weren't great quality. And I was like, I would be really shocked if this isn't hop latent viroid. They got a test done and all of them came back negative. And I was like, I don't understand that at all. And then my question was, did you take from multiple parts of the plant? Did you take from the roots? Because the roots seem to be the place where the virus hangs out the most, right?
It prefers to head down to the roots. It kind of follows the flow of nutrients, phloem material, sugars, and so on. They end up going in the roots and then the plant redistributes it. And so it's common for many viruses, including the viroid, that they follow this pattern. But usually, when you've got the symptoms on flowers, I've noticed that you can detect hop latent viroid in those flowers as well. So it's not a matter of the roots are diseased, but the flowers are not, and you get reduced THC. The flowers also get infected. So
Mm.
that test should have been positive, unless the cause is something else, right? There's another
Yeah,
issue
and I
out
can't
there.
I mean I'm not a plant pathologist, but I couldn't think of another disease that would
Well, that,
result in every single, and I don't know if it was multiple strains or just a single strain. I don't know if they were sharing a water source. There was like a lot of questions that I had that they didn't answer. But if you saw that, like what would you, and they came back with a negative, negative test for hop latent, where would your brain go next to
yeah, so we're,
investigate?
we're actually starting to see that a little bit with some of the farmers or the growers that I work with now. I think we meaning the people I work with have solved the hop latent viroid problem because basically what they did is they culled out every single plant that was infected, especially the mother plants. They just completely got rid of them and they put in all the sanitary practices, no movement of plant material, everybody wears gloves, etc. But yet we're still seeing a little bit of a reduction in some plants with THC. So for example, 3-4%, which is not as significant as hop latent. So I'm thinking now that maybe there's another virus disease that we haven't really quite looked at yet. And so now we're doing, what we call this whole genome sequencing thing with RNA, which is what most viruses have in them. And it's still a little early to say, but we are seeing sequences that don't belong. There's sequences of something in there that resembles a virus or viruses that we still have to give names to and find out. So there are things happening inside these plants which are vegetatively propagated that have been for so long that they're keeping some of these unusual pathogens, perhaps viruses that eventually may start causing problems on these plants. So we're not done yet in terms of research because there are things in plants that may be causing these small reductions in yield, and viruses tend to do that. They kind of eat away at the plant. 3-4% may not sound like a lot, but if you've done it for 3 years in a row, you're up to 12% reduction already. So I think the story will continue in the next 6 or 8 months. We may be able to put something out that says we've found another new virus in cannabis. And it's not unusual because you see that in humans, right? As we propagate and evolve, new viruses appear out of nowhere.
Someone was asking me, it was Bill Graham. I don't know if you're familiar with him. He does micropropagation, but he asked an interesting question, which I didn't have the answer to. So these viroids, they're really small pieces of naked RNA floating around doing damage. What if they're naturally occur, like they didn't come from anywhere, but they naturally, develop in these plants, or they were always there, dormant, and at the right time, they kind of flourish. Is that a possibility or
I mean, it's likely, although we don't see a lot of homology with the viroid to any pre-existing part of the plant. So in other words, there are theories about chloroplasts having evolved and or certain organisms having evolved from chloroplasts, and you can see similarities in DNA sequence. The viroid doesn't show any similarity to any part of the plant that we could say, look, maybe it originated from this particular location. So the origins of those are really interesting.
Mm,
What's more interesting is that you don't see that in animals. You don't see viroids in humans or any other type of animal, pretty much just plants. So maybe they evolved many, many years, millions of years ago for a purpose. And then
hmm
that purpose either went haywire or the viroid decided, we need to start causing some damage here so we can reproduce and spread. And then that's maybe what happened. I don't know. The whole history behind the viroid is so unusual because it's such a tiny, tiny piece of RNA, like
hmm
super small and stable. Like, we ended up autoclaving some material that was had hop latent on it, and it came back positive. Like it was still viable. The RNA hadn't broken down even after one cycle of autoclaving, which is 121 degrees Celsius and 15 pounds per square inch pressure on a normal cycle. So that thing is almost, I'm not going to say totally immune, but it's pretty resilient.
Which is weird because I was always told that RNA is not stable. And when you're doing things, like looking at the transcriptome and you're taking the RNA for the snapshot in time of whatever that organism's doing right then and there, like you got to work quick because the RNA has a tendency to break down. So it's weird that
Yeah,
that.
it's weird and it's not. I mean, we call it single-stranded RNA, but in reality, what it does is it loops up on itself. It forms these loops that
Mhm.
are formed within the molecule itself. So it's actually more a double-stranded, or it's single with double-stranded pieces. And apparently that makes it completely resilient to enzymes because when the enzymes come along, the RNase enzymes, then they try to chop it up. They run into one of these loops, which is pretty much double-stranded, and it stops. It can't get beyond that. So that double-stranded wrapping
Yeah.
is what seems to be making it super, super stable.
So it's like single stranded RNA all doubled over in the fetal position so nothing can get at it.
Yeah, pretty much. It's got its barriers up on the outside, right? So there's a bunch of these loops, I don't know, maybe 8 or 10, and that apparently really adds stability and it makes it more of a rod shaped as opposed to sort of a free RNA or circular RNA. It's more. So yeah, the stability is incredible. I mean, we know there's RNA enzymes that will break it apart. And so we kind
Mhm.
of did a small study. Could we use the RNase enzymes as a way to manage this disease? But you'd need a lot of it. You'd have to get it right into the plant, into the root zone, blah blah blah. There's certain bacteria that actually produce RNases that perhaps could be looked at. So we're looking at that as well. Do we have naturally occurring bacteria that produce RNase enzymes that could break down the hop latent viroid? Now, you can do it in the lab, and then the question is, how do you get the bacteria everywhere into the plant, right? You got to get into the roots, you got to
Yeah.
get it into the flower. So that might be more problematic because when hop latent viroid gets in, it's completely systemic in the sense it's
Yeah.
it's throughout the plant. And so I don't know how a bacteria could chase this thing down, but in theory, there are enzymes that will break it down.
I feel like that's such a human thing to do to try and, fix one problem and then create another problem though. Like, you introduce a bacteria and now we've got another problem with
Yeah, you got to worry about yeast and mold and bacterial levels in
yeah.
in the flowers as well, right? So yeah.
Yeah. So So, yeah, hop latent viroid issues. And so another pathogen or, issue that I find interesting is the mosaic viruses and natural variegation. I've seen people who say there's no such thing as natural variegation, it has to be a virus. And I'm like, why wouldn't that's not true. Like variegation is, the lack of pigment in and that of course can be created from a mutation, right? The
Yeah.
mutation in the thing that creates the color. But the mosaic viruses also create that modeling pattern. And I see everywhere still people talking about tobacco mosaic virus. And if they see a mosaic pattern on a leaf, they're, oh, tobacco mosaic virus. I have heard that there have been some positive tests, but like I've never been able to get anybody to actually show me a positive test for tobacco mosaic virus. And I think that for the most part, everybody is way overstating the prevalence of tobacco mosaic virus in cannabis. If it does occur, I I don't think it's going to lead to like huge problems. I don't, like people are like, don't smoke around your plants. Don't put your tomatoes anywhere near your cannabis. You're going to get infected. I want to hear your take on tobacco mosaic virus specifically in cannabis. There are other mosaic viruses too that do infect cannabis, but tobacco
Yeah.
mosaic is not
So,
really
so
one of
again, going back to the days when I started, we saw a lot of those mosaics, and we were convinced that, and we were convinced only based on the internet searches that we did, where they showed exactly the same symptoms and said tobacco mosaic, tobacco mosaic. So we went around thinking, okay, this could be it. But as you said, it wasn't spreading. Anything we know about tobacco mosaic will tell you that it really spreads a lot. And so
them.
if you had one plant showing this mosaic, we wouldn't see necessarily spreading to another. And also the mosaic was localized. Certain leaves had it, other leaves did not. In
Hmm.
tobacco mosaic, it does the same as hop latent. It becomes systemic, and so
Hmm.
the entire plant will be affected. So the distribution of that mosaic was not uniform and it was not spreading. But we ended up doing again one of these RNA sequencing experiments where we took the entire leaf, we ground it out, and we ran a full genome sequence on it. And lo and behold, we came up with nothing. We
Hmm.
found nothing in there that would tell us in those particular plants that we had a new virus or tobacco mosaic or anything like that. So we then said, okay, it's got to be a mutation. Those are quite common. And when you look at it really carefully, some of it, the mosaic is only on one side of the mid vein. It goes this way, which is typical of mutations.
Hmm.
Cells kind of divide on one side, not the other. Tobacco mosaic virus wouldn't do that. It would kind of spread everywhere.
Yeah.
But we're still going back to that though, because I think, as I mentioned earlier, with the 2-3% reduction in THC, that there may be these viruses out there that are in the plant that may not be really obvious in terms of mosaic and other patterns, but that may be causing some kind of issue. But I would say right now that tobacco mosaic is not one of them. It's not one of
Yeah.
the ones we need to worry about. And in fact, there's very few viruses on cannabis. When you think about where it's grown around the world, what, five, maybe six viruses have been described, and some of them are from Israel and haven't really spread too far beyond that. We've only seen a couple of three, beet curly top. We've seen maybe the mitovirus, which doesn't seem to be doing anything. Yellow leaf curl is here and there. So there's not a lot of viruses. When you think of tomato, peppers, potato, any other crop that's been grown for a while has a lot of viruses. And I don't know the reason for that, whether it's got some natural resistance or maybe it just hasn't got to the plant yet. Maybe we haven't, thank God, we haven't seen the viruses take off.
Yeah. Well, so alfalfa mosaic, beet curly top, lettuce chlorosis virus, right? Those all infect. Then there's isn't there a hemp
They used
and
to call it hemp streak, but then I think they changed it to something else.
then there's cannabis cryptic virus.
Yeah, yeah.
That's
So
the best name they could come up with. Like we don't
yeah,
know virus.
yes, yeah. So that's like 4 or 5. That's not a lot, which is
Yeah.
good news. I mean, when you think of a plant that's propagated vegetatively, and there's so many growers handling these plants all the time, right? You're constantly pruning and taking leaves off and stuff, and we still don't have a lot of viruses. So that's good news.
And it's super interesting because We think about cannabis, we bring it inside and we give it all the lights and the nutrients, and it has a reverse osmosis water, and we're wearing gloves and hats. Like, we really do baby this plant. It's amazing that this poor plant can do anything at all for itself. It's like,
But boy, it sure grows well though, eh? But when you fertilize it and stuff, you come back in a week and that thing's already grown another 8 inches or whatever, and it's amazing that you can cycle it in 3 months and get what you want out of it. It's fascinating. It really is.
yeah, yeah. Well, we jumped right into viruses, but you know what? I didn't even ask you. You are up in Canada. You're at Simon Fraser U University, and I know that you've worked a lot with U.S. cultivators and stuff. But like, you're a plant pathologist, you're an educator, academic, and you talked about how you got dragged into cannabis. But um, like, I'm sure you do. You still work on other plants, or is cannabis really your focus right
Yeah,
now?
so I mean, I've got a small project on tomato right now. I've done some work with blueberries. Basically, I try to focus on crops that we have in this area in Canada that for which there's funding. I mean, as you know, without funding, we can't go very far. So there are other crops. I did some work on wasabi. We found an interesting virus on that one. And then the other big crop I had about 10 years ago was ginseng. We had a
Mm-hmm.
huge ginseng industry and we were exporting stuff to Hong Kong and parts of China. And that kind of ended when they started growing their own ginseng. So I've fluctuated a lot between different crops. I would say after ginseng, my favorite one is definitely cannabis because
Yeah.
there's very little work done on it and there's funding. We got funding from both the federal government and then we get funding from the private industries that support us. So it's been good that way. That keeps students going, it keeps technicians going, and keeps me going.
Yeah, I mean, it must be nice living in Canada to be able to work on Canada. I need to come up
Yeah,
and work in Canada.
but it's I get butterflies in my stomach when I enter the US because the last thing I need for them is to ask me, "Hey, what do you work on?" I worry a little bit sometimes because of the differences in regulation between Canada and the US, but as you said, I work with a lot of US growers, producers, because they want to know what diseases we're seeing and they're seeing the same things. They're seeing Fusarium, Pythium, powdery mildew, of course, is everywhere. And then
Hmm.
hop-latent viroid, which really started, or at least was discovered first in the US. And then we found it here in 2020. So a year after it showed up in California. And again, we move plants back and forth all the time. We got hobby farmers, growers go down to the US, they pick up a few things, bring it back. And so we introduced it somehow into Canada a year after it was found in California.
I mean, nobody knew about it. They didn't know that they were moving something
Yeah.
around, and and Jeremy Warren of Dark Heart Nurseries, they've since have joined up with, I think, Curaleaf. He's really the person, I think, correct me if I'm wrong, that kind of figured out hop latent viroid. And that was back in what, 2019?
Yeah.
I've heard him tell the story, but he doesn't do podcasts anymore. So, can you tell the story of hop latent viroid and how, it was figured out, and then the prevalence and sort of, from then up until where we are now with it being pretty much everywhere. I like to tell people it's not really a matter of if you get hop latent viroid, it's a when,
Yeah.
right?
Yeah. Yeah. So, I mean, the story goes back. Well, it was first discovered in actually two different labs. One in Jeremy's lab and the other one in Oakland. And they both presented a paper or an abstract that said that hop-latent viroid is established in California. And then the source obviously must have been hops because that's really the only other natural host of it, although now we know there's other experimental hosts. So somehow from a hop plant or a hop farm, through touching or through whatever means, the viroid ended up infecting cannabis plants. And then
Mm-hmm.
because it's vegetatively propagated, and when we even do tissue culture, if we do tissue culture just using cuttings, not the meristem, but just the cuttings,
Mm-hmm.
uh, it will propagate. The viroid still goes through tissue culture. So that would be one very easy way for it to spread. And then, of course, distribution of plant materials all over, including up here to Canada. Now we know that if you actually take the meristem, which is the tiny, tiny, tiny piece up at the top, you have a pretty good chance. Not 100%. You get about an 80% chance that you've cleaned up the plant from hop latent viroid. The only way you can actually prove that you've got a good, clean plant. Otherwise, you're spreading it through propagation. And again, because you can't see symptoms, it makes it more difficult. So I think the
Yeah.
spread primarily was just through dissemination of propagation material that had the viroid that nobody really knew about uh,
Yeah.
until the tests, and then tests became available, right? So now you can
Hmm.
run your PCR test literally overnight. So you can
Hmm.
send a sample off, and the next morning you know whether you've got the disease or not.
Right. And so talking about cleaning with tissue culture, I've also heard, multiple opinions on this, that it is possible, and I believe that there are people who've had this happen where they do a cleaning, they get back their tissue culture, it's disease free, it's been tested and no hot latent viroid found. Okay, good to go. And after several runs, the disease shows its head again. And so I'm wondering if it's kind of like, if it's possible that it's kind of like, viruses in humans that, like chickenpox is a great example. Once you're infected, it stays, it hides out in your neurons where your white, your immune system doesn't find it. And then when your immune system is down, you get a cold sore or you get
Yeah.
shingles.
Yeah, I mean, it's possible. Although I tend to think that a lot of it is just reintroduction through either careless or unknown sources.
Hmm.
I know we can get rid of it because, as I said, some of the growers we work with, where they went absolutely wild on cleanup and
Yeah.
making sure that everything was done the right way, and then they started with tissue cultured plants. We don't see that viroid anymore. So there's hope. There's hope that it can be cleaned up. I say 80% of it is clean. Some of it could still have a little bit of viroid in it. And then when you propagate it over 3 or 4 cycles, boom, all of a sudden it does show up again. Because,
Yeah.
the tip that you use to collect the plant material, usually it's like 2 millimeters in size. If you use larger pieces, you may still keep the viroid in there.
Mm-hmm.
So it's not foolproof.
I could see a situation where your viral load is so low that it ends up being non-detectable. I mean, PCR is pretty sensitive,
Yeah,
but you can get false negatives. We know
yeah,
that.
yeah.
Unlikely to get a false positive unless you've got some sort of contamination issue in your lab. But, people who do good testing are going to run a positive and a negative control to make sure that they're not
So
getting.
you should know what's going on. I don't have a good sense now of how prevalent the hop latent viroid is in Canada and the US. We're still doing some stuff on it, but because the grower that I work with doesn't have it anymore, we don't consider it a problem. So we've stopped doing some research on it, other than perhaps looking at the effect of different genotypes. But I'm sure
Mhm.
it's still out there. You may know more than I do as far as growers complaining about still seeing hop latent viroid. I'm sure it's still out there.
Oh yeah, totally. It's totally out there because, like I said, I think that some growers either are not concerned with it or they don't think it can happen to them, like with a really good integrated pest management program. I don't know. I feel like it doesn't, it's, you can be as good as you can be, but humans are humans, right? And you could
Yeah,
bring in a cutting that has it, and if you're sharing a water source and a tray of clones, now all of your clones have it, and you put them in different rooms, or like you very easily get out of control pretty quick. And so, I I don't know if this is, is my advice for growers would be to have a proactive testing as part of their IPM, that they test early, test often, maybe at the beginning and closer to flower, just to make sure that if there is a problem, you can kind of cut it off at the pass and not waste time trying to grow out something that is potentially not gonna give you the
yeah,
yield or
and
the numbers
the
that
other part of the bad news is that I have to tell the grower, you need to get rid of these plants. Like you need
you.
to
Hmm.
destroy these plants and the mother plant that it came from. And that's
Hmm.
really hard for them because they've invested all this money in there and they don't see much happening to the plant. It's not visibly dying. And so that news of destruction is not easy to take,
Hmm.
particularly if it's a mother plant or source of genetics that you've had for a while and you don't want to part with it. And so they
Right.
kind of hang in there. They try taking cuts from the top of the plant or do various things to minimize the transfer. Once it's in the plant, it's pretty much everywhere.
I think proactive testing is it's just good practice because it can be brought in not just from, cuttings from a nursery. It can be brought in on, you know, dirty tools or,
Yeah,
all, seeds even can carry the virus
yeah,
now.
yeah, and you know what? It's money well spent. When you think about all the amount of money you've placed into making those cuttings root and getting them into the veg stage and so on, somebody says I'm gonna charge you $50 for a virus test, a viroid test. It's insignificant compared to what you would have already paid to keep that plant going. So, the test now at 50 is pretty high. I think now they're down to 30 or less per sample, which is not a lot in terms of the bigger picture.
Yeah, MyFlora just came out with a proactive test that tests for hop latent viroid, Pythium, and Fusarium. It's like obviously you'd want to test for hop latent viroid, but we throw in the other two just because they're pretty prevalent as well. All three tests is only $18.
Yeah, yeah.
And it's to protect your crop to give you some sort of insurance. I think that's a pretty dang good price point for a
It
cultivator.
is.
The idea of tissue culture banking, I think, is a really good where people have their mother stock in tissue culture, in holding, and not necessarily having their whole room come from tissue culture, but having their mothers switched out every few cycles or every couple of years to make sure that they've got clean stock and they've got a backup all the time. Because, when you lose a mother, that's devastating, you
Yeah,
know?
so I do like that idea, and I've seen some growers do that. They bank their plants in tissue culture, keep it for, they get charged a certain amount per strain. It'd be interesting to see what those plants look like when they come out. They should be identical,
Hmm.
unless there's genetic changes occurring in tissue culture. So if growers can afford it and there's a facility that does it, I think that's a good idea.
Hmm. Yeah, I really like that idea. I think that it's just a good way to ensure that you can keep going with.
Well, and you're not constantly propagating, right? That's where some of these mutations come in. There
Mhm.
was papers, I forget, a couple of years back. I think it was out of Eastern Canada from Quebec. And they showed that each cycle of propagation creates all kinds of somatic mutations. The numbers were in the hundreds.
Yeah.
Hundreds of these mutations. You don't see them all, but they're being created through this cycle of propagation. And that's where, in some ways, seed production is good because in seed production, you're leaving all those mutations behind. All the
Hmm.
all the ones that are in the somatic cells stay behind. But then of course you got to deal with genetic variation through
Yeah.
meiosis and these other things. But
Yeah. So when I was doing my research at UNC, I was wondering about, what they call genetic drift. It's not genetic drift. Genetic drift is an evolutionary process that's at the population level. You wouldn't see it in a grow that's being propagated through cloning. So correct the language out there, the cannabis industry. But when you see changes over your generations, your clonal generations, I was thinking, well, it's not the genome that's changing, so it's got to be something else. So either mutations, which is totally, plausible, but I was thinking about stress and epigenetic changes to deal with that stress because, plants can't get up and move, and one of the things that they have in their toolbox is to epigenetically change things. And if it's really bad, like it can be an epigenetic response to stress, can be like a Hail Mary. Like I'm just going to throw everything at the wall and hope something sticks to make it through to the next generation. It's like set some seeds and then, and so I'm thinking, well, every time you cut a plant, not only is the mother going to feel the stress from being cut, but those little plants that you now are a whole new individual, they have to make roots and they have to grow and they're now an individual away from mom. And so both of those things are undergoing some sort of stress level. And I can only imagine that there is some sort of epigenetic changes in both of those that can lead to phenotypic expression changes,
yeah,
like differences in terpene profiles, like if it's experiencing being cut to the plant that feels like it's getting eaten, right? So it's going to upregulate or downregulate, something that they think is, that is going to help it survive through this mastication that it's
yeah, and,
perceiving.
and and that's interesting because again, for the listeners that may not be familiar with epigenetics, I just finished teaching my first year bio course, which is a big class with 300 plus students, and
Hmm.
we talked about genome changes and so on, and so we talked about epigenetics for maybe 15 minutes, and I gave them examples of where certain characteristics of humans are affected by diet. They're
Hmm.
affected by stress in the environment. And what basically happens is that in epigenetics, your DNA perhaps isn't altered because you still have your intact DNA, but the expression of certain genes in your DNA is affected. Either
Hmm.
the expression or yeah, basically the expression levels may be reduced or in some cases may be higher. And that happens through environment, through various types of stresses. And sometimes they are carried forward into the next generation. So for example, mothers that are pregnant that have kids that are totally stressed for whatever reason, you may start seeing behavioral changes or psychological changes in the children. 15, 20 years later, they've shown in humans that this can actually happen. And it's all epigenetics. So in plants which are propagated every few weeks, those changes could be numerous and carried forward into following generations. But certainly in humans, it's becoming a very interesting area of research. Now, there's not a lot of data right now to demonstrate that, yes, these are true changes. They've looked at the DNA sequences. Many of them are similar, but the expression of certain genes can be up or down regulated.
Yeah, so I did a deep dive into this and, you know, animals versus plants. So plants cannot move, right? They can get up and walk away when shit gets real. Animals and epigenetic changes, generally those markers are wiped away during meiosis. There's mechanisms to wipe those things away so that you're not passing on, something that might be deleterious in terms of an epigenetic change to the next generation. You really want to wipe that slate clean. Doesn't always happen, but for the most part there are mechanisms in place. Plants, on the other hand, have a tendency to carry through epigenetic changes because if that plant is undergoing some sort of stress, drought stress or whatever, and they're able to make a seed, and that seed is presumably going to stay in that environment, which means that it's going to have to grow up in that same stressful environment, though it's very advantageous for that to carry through the next generation so that they can survive that environment. And so
Yeah,
I was like super interested in that because plants seem to carry those epigenetic marks through multiple generations.
yeah. Yeah, no, that's fascinating. Certainly, it's an area that needs more work, particularly if we find epigenetic changes that are beneficial. So, under high temperature, under high salt stress, they do better. That would be a nice change to move forward.
Yeah, and these epigenetic changes can become permanent too. Like
Yeah,
it does become part of the evolution and adaptation of that organism to
yeah.
whatever it changed due to, I guess.
And
So
cannabis would be an ideal plant because you can vegetatively propagate it for X number of cycles, and then start checking to see what genetic differences there might be as a result of, or put them under different stress levels and see what
I
happens.
grew up mothers and then I cloned them and then I took, cuttings from the mother before and after and then from the clones and then I did clones of clones and clones of clones of clones and then clones of clones and all I was going to do because I had no money to do any research, so I had to pick the really cheap alternatives. And so we did some methylation sensitive digests to see if to see what methylation changes they were.
Yeah.
So for people who don't know, one of the epigenetic mechanisms is that a methyl group attaches to the DNA, and there are enzymes that will cut those sites where the methylation has attached. So whether that it's been methylated or methylated, you would be able to see those changes from generation to generation. And so that's all I wanted to look at is there changes? That's all sitting in a drawer still. But there's a ton of samples to run, and I'm hoping that maybe an undergraduate or a master's student will come and pick that up because I think it would be a really interesting thing and
Yeah,
not
I
too
think
expensive to do.
no, no, and it would reveal some interesting insights into what this plant is able to do.
Mm-hmm.
Yeah,
Yeah, because I think of back in the day when you'd make a mixtape, and then somebody wants a copy of that tape, so you make a tape of the tape, and
yeah,
then a copy of the tape, and it just kind of like keep the quality of the
yeah,
recording just goes to shit. I kind of feel like that's how it is with clones, and then clones of clones, clones of clones of clones.
yeah, for
Yeah.
sure. Yeah. Okay,
All right. Oh, I wanted to talk about my last time I looked at your publication list, seems like you've only published one paper this year.
okay.
Let's hear about this research
That
because it's
that's
pretty
that's
interesting.
really that's one that I thought was quite interesting because it happened accidentally. And so while I was doing my walkthroughs in some of the growers' facilities, I noticed some plants of one particular genotype or strain were acting really strange. The leaves were starting to wilt, and I could see kind of white patches on the stems. And I looked at that and I go, that's really odd because if it's powdery mildew, it very seldomly you see it on the stems. It's on the leaves and so on. And it was an intense white, like the same color as your sweater. It was just really white or whitish beige. So it's got to be a new pathogen, I said, because that's how my mind works. So I collected a whole bunch of these samples, I brought them back, did the regular isolations. Nothing grew out other than penicillium, which is everywhere. Then I thought, okay, we've set up some experiments with DNA sequencing, so let's throw some of these samples in there. Maybe it's a completely new pathogen because that would tell us what DNA is there. So I took, little leaves that had showing these sort of patches of wilting and whitening, and send them off for DNA sequencing. So then I said, well, use a hand lens or a microscope. Maybe there's something I missed. And I looked in there and I saw crystals. I saw these crystals in there. I thought, wow, that's fascinating. I wonder what's going on. Is something dripping on the plants? I looked up. Maybe there's, something dripping and burning the leaves. Nothing. So then I thought, well, now what's going on? Well, let me use a scanning microscope, and I've used that before to look at these glandular trichomes, because they're so big, you could see these balloons forming. But now there are no glandular trichomes on these leaves, right? These are leaves and stems. So, but they got lots of hairs, it's just covered in hair, which when we touch is what kind of makes us itch or causes
Mhm.
rashes. So I looked at that under the microscope, and what I observed was that these crystals that I saw with the hand lens, they were sort of aggregating around these leaf hairs. These trichomes were accumulating these crystals, and when I looked even closer, I realized that these leaf hairs, and they're pointed. They're nothing like the glandular trichomes. They're skinny and they're thin. They're sharp. At the ends of
Mhm.
these skinny, sharp trichomes were little drops of crystals coming out. Literally, the plant was forcing through this salt. We measured the EC, the solution that was the plants were in. It was super high. It was over 10, which normally is like 5 times what you would normally grow on cannabis. So what was happening was these plants were getting over fertilized, or there was something was blocked, and their plants were just accumulating high levels of salt. So they were showing symptoms. There were patches of white and brown. But what was even more interesting was the fact that they were pushing these crystals out. The salt was being taken out and it was accumulating on the surface of the leaf. So then I thought, that's interesting. So let me read more about salt in plants. So there's a whole bunch of stuff on salt in plants. And there are plants that are called salt tolerant. And salt tolerant plants will take salt from the environment picked up in the roots and they'll expel it. And the reason they'll expel it is that it doesn't become toxic to the plant. It's like when we perspire,
Mm.
we're exuding things out of our skin pores. But these plants were taking these salt levels and reducing them by pushing them through these. So that's the mechanism is very similar to a plant like mangrove, which grows in sea salt. It grows in
Mhm.
seawater. And if you look at the leaves of mangrove, they're white. The outside crystals. The sea salt is just being pushed out, so it's not toxic to the plant. Now, cannabis or hemp, as we know, is pretty tolerant to environmental stress. It can grow in desert climates, it can grow in areas that probably have a lot of salt in it. And we know that they accumulate salt, right? They're hyperaccumulators. So maybe, and this is just a hypothesis, maybe one of the mechanisms these plants have to deal with excess salt or fertilizer or whatever is they take it up. They can't not take it up. It just kind of goes through the roots. But then when it gets up into the leaves, before it becomes really toxic, they push it out. They send it through these leaf hairs and they start accumulating these crystals on the outside, which could be a mechanism for tolerance to salt in cannabis and hemp. But
Mhm.
at least it gives us or gives me a reason why these plants have these hairs, these trichomes. They're great for insects, they're great for maybe deflecting light, but now we
Mhm.
think that they may be great in also pushing out excessive salt from these plants and making them more tolerant to the environment. We have a purpose now for why nature made these plants so hairy.
Yeah. Well, and trichomes are not exclusive to cannabis.
Oh
Lots of
no,
plants, lots
almost
of plants,
every plant.
tomatoes,
Oh,
tomatoes
tomatoes covered.
covered,
Cucumber.
right? So all
Cucumber.
those little hairs that on any hairy plant, right? Those are all trichomes. So do you think that like if you were to do the same thing with a tomato, those tomato trichomes would also push out salt?
It's possible. I know it happens in cucumber. So in cucumber, when they flush the plants with silicone, they use potassium silicate to control powdery mildew. So they flush the entire plant with fairly high levels of silicone. And so by
Those
pushing
little
these
pokey
salts up,
bits,
it's not really white, it's a blush. It's almost like a wax cover on the leaf, on the fruits themselves.
like a bloom.
Yeah, like a bloom.
Okay.
You got it. So it does occur in other plants, and presumably, plants don't want a lot of silicon. They want some. They're pushing the rest of it out. And so I had a cannabis grower that was using silicon as well. And he says, I got to use it because I got powdery mildew everywhere. He called me back a week later and said, my flowers are turning white. I go, what? He goes, yeah, I'm looking at my hand lens and I'm seeing crystals on my trichomes. And I'm thinking, boy, that's got to be silicate again. So he already had observed these things coming out of the plant in the flower tissues because he had fertilized with excessive amounts of potassium silicate.
You
So
don't want that on your flowers, right? If you're
no, you don't want that. He was concerned
yeah.
because it almost looked like mildew, right? It's the flowers were literally white. And I said, that's got to be crystals. And sure enough, when I talked to him, he was using that. So he immediately stopped, flushed his plants with water, and those crystals went away. But it proved the concept that because flowers do have the hairy trichomes as well. We got the glandular, but you've also
Hmm.
got the what I call leaf hairs, and
Hmm.
the crystals were coming out of the leaf hairs, and they were just precipitating out. You see these clusters of crystals that nobody's ever reported this. For me, it was a breakthrough. Yeah,
Interesting.
I mean, the practical applications, others will have to pick up on that. So the next experiment would be to take these strains and grow them with different levels of salt, high,
Hmm.
medium, low, and see whether or not we're seeing differences in the excretion. I call it excretion of salt out of these plants. No.
But more as like because we know cannabis is a bioaccumulator, maybe a desalination remediation kind of like
Uh, yeah,
environmental
yeah,
remediation where they need to get the salt out of the soil and potentially use something like cannabis to suck all that salt
yeah.
out of the
Yeah, and
earth.
we know from early literature that things like cadmium and lead and copper that's in the soil can be taken up in cannabis plants. But surprisingly, there was only one study which actually showed, and I think it was copper, that these copper actually ends up in the trichomes. Before
Hmm.
they said, well, the plant takes it up, fair enough. But now it's coming in through these particular leaf hairs. And so they may have a role not just for general salt, but also for heavy metals.
Hmm. Yeah, or yeah, anything that needs cleaning up.
Yeah, and then of course you don't want to eat those plants or use them for anything else. They're just there for the purpose of remediation.
Hmm.
Anyway, yeah, so that's been published recently, and it's been published online, but
Yeah.
the actual article should come out in the next month. And just so that you know, for the record, I have submitted two more papers in February. I've got to keep that track record happening. So
You've set that bar really high. If you can't you can't stop now.
Okay, we'll see how that goes. But anyway, it's all because I like to write. That's the main, and well, and cannabis is fascinating.
Yeah.
That's
Be-
fascinating.
Yeah. And there's so many subjects to publish on to and you seem to have a lot of things that you get to observe and write about. Make
Yeah,
that bar
and I can
be.
do studies, right? I can follow up and I
Yeah.
can take these samples and analyze them whereas a lot of people can't. So I'm lucky that way.
Mycotoxins.
Oh boy. Okay.
I mean you're not a toxicologist, but in your opinion, so this has been one of the big discussions as of recently is are we testing the right kinds of what do I want to say? Are we testing the right kinds of fungi and molds and things like that on cannabis, like in terms of human health, or are we just like, I don't know. I don't know how to ask this question. Maybe you
Yeah,
can.
I think I know what you're getting at. So
Yeah.
if we start with mycotoxins in general and think about how mycotoxins are produced in other plants, and I think wheat is probably a good example because we see a lot of it in wheat. There's about maybe five fungi that we know are, or molds that we know are good mycotoxin producers. And one of them, of course, is Fusarium. And then you've got Aspergillus, and then you've got Penicillium. There's also evidence for Alternaria to produce mycotoxins, and in some cases maybe botrytis. So you've got the sort of the deadly five that you'll find not just on wheat, you'll find it on apples, you'll find it on tomatoes, any crop plant that allows the development of the mold to the extent that you can see it with your eyes.
Hmm.
We have to distinguish what you see with your eyes compared to what is found in a yeast and mold test. A yeast and
Hmm.
mold test will tell you that you've got, 20,000 colony forming units, whatever, but you don't
Hmm.
actually see it. It's embedded inside the flower. When you actually see mold with your visual eye, whether it's white or yellow or black, to me, that's an indication that you're going to start seeing production of mycotoxins. So they only tend to be found in under conditions where either your cooling system is broken down, your humidity is starting to go up. If you're growing outdoors, there's been a heavy rainfall
Hmm.
closer to harvest, and now everything's turning moldy. That's when we'll start to see mycotoxins. Finding yeast and mold in flour doesn't mean that you've got mycotoxins because those levels tend to be quite low. So we have seen mycotoxins with Fusarium on cannabis flower in the greenhouse. I don't know how it got there, but the flower structures were turning white or pink, like you would see on wheat. So under those conditions, we actually did find mycotoxins for the first time, Fusarium
Hmm.
mycotoxins in cannabis, the same ones that occur on wheat. And clearly, if that product was consumed, it would cause problems for people. Now, fortunately, it's so moldy that when the thing started going on the conveyor belt while they were sorting flour, they were being picked out. People that were saying, "Oh, this one's bad, this one's good." They started picking these things out. But theoretically, some of them could still get into a product, and that's when we need to be really careful. So Fusarium mycotoxins, yes. We know about Aspergillus, that's already on the testing list. We know about Penicillium, ochratoxin, it's on the list. Alternaria surprised me because we found Alternaria toxin as well in these flowers because they had Alternaria. They were kind of turning blackish. Looked like botrytis, but it was actually Alternaria. So cannabis and hemp are hosts for these fungi. They can produce mycotoxins under these humid, wet conditions where flower is turning moldy. Um, should we be concerned? Yes. We need to develop testing for other mycotoxins that are not on the list, and that would include all the Fusarium ones and possibly Alternaria. We know that we have the Aspergillus and the Penicillium ones there. But surprisingly, when you look at the literature, there's not a lot of reports that confirm a lot of mycotoxins from Aspergillus in cannabis. There are reports, a few. Perhaps the concern more is breathing in Aspergillus spores,
Hmm.
because then it creates problems with your lungs and so on. But mycotoxins from Aspergillus reports are very few. I would
Yeah.
say right now there's more reports of Fusarium mycotoxins than there are reports as in people discovering and seeing more. But those are not being tested for.
So kind of the regulations should be adjusted to be more, because from what I understand, the regulations really come from the food industry, but
Yeah,
we
historically,
don't
yeah,
eat cannabis. We light it on fire
yeah.
and in some cases concentrate it. And so yeah, I feel like the regulations maybe need to be adjusted to be
I
more,
think so.
yeah. I've heard, there's been a rise in CHS, cannabis hyperemesis syndrome, which, hasn't been pinpointed the exact cause of that. Almost all arrows point to it being, an overload of the endocannabinoid system, kind of tipping you past a point where you end up getting ill. Folks like Tess Edom are concerned with vomitoxin,
Yes.
and that
Yeah.
we're not testing for the right things that could be making people sick, and it's getting misdiagnosed as CHS. And I don't know where vomitoxin comes from, or do you know much about that or no?
Yeah, so that's one of the ones that Fusarium graminearum produces. That's one of the ones that is a big problem in wheat scab, and we found it. We found
Mm-hmm.
it in cannabis flowers, and naturally occurring, but also we experimentally added the Fusarium to the flowers because there's a lot of non-believers that, oh no, cannabis can't get mycotoxins. There's no way. We wanted to prove that it could. under experimental conditions, take some Fusarium, put it on a flower, stick it in a plastic bag, let the mycelium mold get covered, test it. So
Hmm.
we found the major ones that are found on wheat, and then we found the alternaria one. We also found the penicillium one, mycotoxin, but we never found any aspergillus, zero. That doesn't mean it's not there, but you would think under those humid conditions in that bag that we'd also see aspergillus. So I'm not saying it's not important. I'm saying there's other ones out there that we should be more concerned about as we start growing more and more hemp and cannabis closer to wheat fields or barley fields or wherever the
Hmm.
spores are coming from. In our case, there was a barley field quite a ways away, but I guess the spores came in through the vents and it ended up on the cannabis flower, and I've never seen a cannabis flower turn pink.
Yeah.
It was from Fusarium.
And there's a difference because our fruits and vegetables and things like that, they go through washing processes where you can wash off things like toxins and mold and blah blah blah. In cannabis now, and I think they're doing it in Canada all the time, is remediate, like sterilizing, remediation steps
Radiation.
to
Yeah.
kill various yeast molds, various organisms that might be on your cannabis. That those processes don't get rid of the toxins, right? The
But
toxins will remain.
it does depend. It depends on the dosage. It depends on the duration. When you look at the literature on mycotoxin degradation by irradiation, there's a lot of evidence out there that it does work. If you look at wheat seeds, if you look at flour, if you look at herbs and spices that are infected with aspergillus, there's many studies using both gamma radiation and E-beam. E-beam is what we use here with our growers. I'm not going to say it's eliminated, but it's down quite significantly. There's sufficient studies that show that it does reduce mycotoxin levels, but it depends on the dose, depends on the duration, and so we meaning Canada allows it. It's very expensive, and not recommended unless the market for that product is individuals that have underlying conditions that are cancer patients or whatever, or in countries like Israel and elsewhere that don't allow any contaminated product or mycotoxins in. They have to be completely cleared. And so we in Canada do allow irradiation. I haven't done any studies to show that irradiation will reduce mycotoxins, but I know it completely eliminates mold.
Yeah.
So it works there. But if there's a pre-existing mycotoxin, that's actually my next study. I'm going to be looking at that to see by taking these flowers that I've added Fusarium to,
Hmm.
irradiate those and see what's left.
What does the irradiation do to the trichomes, to the
So
phytochem, all the phytochemicals? Does it change the profile at all, or?
So in theory, in practice, the irradiation actually attacks the DNA and it breaks it down. So microbes that are exposed don't have much of a cell wall like plants do. That
Hmm.
the DNA that's in bacteria, the DNA that's in the fungal spores is completely broken down into little pieces, and that's the end of it. There have been studies to look at what irradiation might do to THC or cannabinoids and terpenes. Not a lot of studies, maybe a couple of three. For the most part, they show that the cannabinoids are not affected. You're still seeing pretty much similar levels, but the terpenes go down.
Yeah.
In some cases, quite significantly. So you will see less odor or less, fragrance that you normally would associate with cannabis in irradiated flower. There are measurements that have been made. There's always a variable, right? When you say
Yeah.
THC, it's plus or minus 3 or 4. Irradiated products are within that range, within the
Okay.
variable range.
I've just heard people in Canada complaining about the quality of the flour, that it's just kind of lost its luster,
Yeah.
and they're
And
attributing it to all of the irradiation that's happening.
I mean, it's possible. It is expensive, and so not everybody's going to use it. They're only going to use it if they've got really bad product, which, by the way, they should not be radiating in the first place. They should just throw it away. But it's mainly for markets where they require it, they demand it, or they demand something that's got zero tolerance. And the only way you can get zero tolerance is through irradiation.
So you're not pro irradiation, but
I mean,
there are certain circumstances where it
I,
is
yeah,
necessary.
I think it's necessary for the cause, for whatever they need, because I'd rather irradiate than run the risk of ending up with spores of some fungus, in my lungs if I was to use it. But on a day-to-day basis, I don't think it's great. I mean, why do
Not
it?
not irradiating because you know you've got contaminated
Yeah,
product and get it to pass, and not irradiating just for irradiating sake.
yeah. And so in Canada, we're fortunate again by having legalization. Every part of the country has one limit, 50,000
Hmm.
CFUs, not 40 or 10 or 100 as it is in different states, right?
Right.
Massachusetts might have 100,000 and Oregon might have 5,000, whatever. We're at 50. I don't know what that 50 means in terms of, I mean, we know it's a spore load, it's how many colony forming units, but in terms of what it can do, our regulatory agencies feel that 50 is fine. It's a doable task. And for the most part, when you grow cannabis well in the greenhouse, you'll very seldomly exceed 50. When you've got growers that aren't following the regulations, or it's a bad day, the humidity picked up in the greenhouse, they will exceed the 50. And they have options of either getting rid of it or using it for extraction. So 50 is a good number that you won't exceed unless it's, a not well-run operation. It's usually
Mhm.
below that. Now, again, I don't know what 50 means. Maybe 25 is better, but we're at 50. We're allowed to run 50,000 colony forming units per gram.
Okay. Good to know for regulators and for people fighting the good fight to get some of these testing thresholds into a reasonable limit where
Yeah, and,
cultivators
and
can
again,
actually.
so we have some growers that use trichoderma. They use it sprayed actually on the flowers to keep botrytis down, and
Mhm.
some of the product fails. It fails because it's got too much of the beneficial spores of trichoderma. So, the yeast and mold testing again is something that needs to be revisited. You may have good mold in there, and yet
Mhm.
you're going to end up destroying product because it doesn't meet the 50,000. So there's a lot of discussion, and I think through various organizations and companies that are trying to do specific testing for specific mold. So only
Mhm.
test for aspergillus, only test for Fusarium and maybe penicillium and others. Forget the rest, and just go after 4 or 5 specific fungi that we know are harmful to humans.
Yeah.
It might be more expensive, and it means we can't do yeast and mold testing anymore. We have to do PCR testing. But maybe that's the way to go, is specifically identify what you want and forget the trichodermas and all the other beneficials.
I mean, I'm a big proponent of doing both plating and PCR testing because the plating isn't going to pick up on dead stuff. So,
No,
here in the United States, you can irradiate, leaving behind toxins that won't show up in the testing. Now you've got flour that's unsafe for consumption,
right.
but is able to pass the test. But PCR will pick up on that, right?
Yes.
It will pick up the dead stuff. So yeah, I'm a proponent of doing both, but that adds a cost
Yeah,
to,
that's
to the to
the
the testing, which is
right.
already kind of hefty.
Yep. Yep.
I
Yeah.
also think that the states or the regulatory agencies should be the ones that are picking up the tab for the testing. I don't, you know, I don't know why they're dumping that cost onto the operators.
Right. Yeah. No, I think the testing needs to be re-examined. And again, this is an evolving process. We're way further ahead now than we were 5 years ago, in
Hmm.
terms of various production practices and disease management and so on. So I look forward to the next 5 years giving us way more information than we ever had.
Hmm. Cool. Well, I'm glad you could shed some light on that because I know that's a big conversation at the moment. I teach cannabis science courses.
Oh, cool.
And I teach basic cannabis science and then advanced cannabis science. And in our advanced cannabis science course, we have an assignment where they have to read a paper. Well, they've got two. They have to read two papers. One of them is yours, your hermaphroditism paper,
Oh, yeah.
which I really like. I thought it was really interesting, but as a geneticist, I'm fascinated, we've got feminized seeds, they're XX, but yet we see a spectrum of maleness. It could look 100% male. It could have a couple of flowers here and there. It can be like totally half and half, like male and female flowers all mixed together. So, and then is it that there's a pathway that's broken maybe, or a receptor that's broken, or like there's so many things that can lead to an expression of maleness in a genetically female plant, which is super interesting. And then there's the idea that all female cannabis has the potential to express maleness because all she wants to do is set seed for the next generation. So like if you let her go too long, if you let her mature too far, the process is rhodolization, where she's gonna stick out some bananas, she's gonna make a couple seeds, and then she'll be good to go for the next season and maybe try again, right? So I would love for you to talk to me about hermaphroditism.
Yeah, that paper was fascinating. It's the only one paper that has gotten three, four, 500,000 views, which is, close to
That's a lot.
half a million is an awful lot. It still continues to be
It's
of
all my
interest.
students.
Thank God for that. The evolutionary biologists and others that work on these kinds of concepts in other plants are fascinated by the fact that cannabis can do both. And so, as you said, we found it accidentally. Everybody sees bananas, the little flowers. We noticed that it was more in some genetics than others. We noticed too that the plants that produce them seemed stressed. They didn't seem
Mhm.
very happy. They may have been at the end of one row. Maybe they were receiving a little bit of extra light. Maybe they were not doing well nutritionally. So there were a lot of things that we felt without ever having proven it, that these plants were experiencing the production of these male flowers. And then, as you said, when you have feminized seeds, which are XX, those plants were experiencing some kind of stress as well. And these male flowers had pollen that was viable. And so
Mm.
it wasn't spreading very far in the greenhouse. It would spread maybe 10 meters or so, but enough to cause seed set, right? So the growers were now coming back and saying, damn it, I've got these tiny little seeds that are forming, and what's going on? And so it was all based on the fact that these plants were doing it. Now, in the article, we talked very generally about stress, that it's pathogens, it could be water, it could be nutrients, but it could also be hormones. There are a lot of different hormones that are produced by plants, particularly one called ethylene.
Mhm.
Maybe ethylene is being triggered to produce these male flowers. So we don't know exactly what's causing it. We also reported on transposons, transposable elements were part of this big picture, just like the epigenetics we talked about. But here you've got segments of genes or fragments that are jumping in and out that are causing genes to be turned on or turned off. They may be part of the story as well, that when they jump in, you get male flowers, and they jump out, you get normal flowers. I don't know. So there's a lot of unknowns, but it's interesting that the plant can do it. It's interesting that it produces fertile seeds from that. We developed a PCR marker based on literature that others had done, where we can now tell male from female plants, true
Mhm.
genetically male from female. And it was really an exciting study.
With a pretty easy PCR test too. It's just
Very
the
easy.
size of the bands. Either it's got the male band or it
Yeah,
doesn't.
yeah, very easy. So it's something that I'd like to go back to. This was
Hmm.
published, I think, in 2022, maybe. I'd like to go back to that work and explore more about what could be causing the female flowers.
I think about people and their stress levels. Some people can tolerate a lot of stress and they do just fine. Other people kind of, want to go to bed and pull the covers up over their head and cry and everything in between, right? So I feel like plants are similar, and there are probably some strains that just they've got a very low threshold for stress and they're gonna try and make us, they're perceiving the end of life at the drop of a hat. And so I'm gonna, make a pollen structure and make some seeds because that's their whole goal in life. Whereas you've got other females that are just like, nope, and you can't do anything to me. I'm never gonna get stressed out. And those are like really good. The ones with the high threshold for stress would be good potential for breeding because you would have a less potential for their offspring to hermaphroditize herm.
And so,
Yeah, so
yeah, so what a lot of the growers are doing now is they're not growing those genetics that they see a lot of the male flowers, obviously. So I'm actually seeing less and less frequency of this phenomenon, unless you've got a favorite genetics that is more sensitive to environmental stress.
mhm.
So some of the ones that I reported on earlier that are not being grown anymore.
I would think that we're actually so for those who don't know, cannabis is pretty unique in the plant world. Only 6% of flowering plants have separate male and female flower or plants, like in separate plants.
Dioecious, yeah.
Dioecious. Most flowering are monoecious, and they have either male and female parts in the one flower or male and female flowers on the same plant. So cannabis is unique in that way. And what we do is we purposefully and almost exclusively separate the males from the females. We do not let them come together. And I feel like, like Jurassic Park, nature finds a way that by doing this, by keeping the males away and by not letting them set seed, I feel like we might be pushing our female cannabis plants down this road of adaptation to start producing more male structures so that they can start making their own seeds.
And that's going to happen with feminized seeds, right? Because that's the
Yeah.
ultimate where you're forcing them to be completely female and produce more females. And there is anecdotal evidence that suggests that feminized seeds do have a tendency to produce more male flowers. But I've seen that in non-feminized seeds as well, or
Mm.
varieties. So it's a bit of both. But the stress thing makes a lot of sense. We just don't know what the stress is or stresses factors. And because that sends out all these hormonal signals that the plant picks up and says, okay, there's a problem here. We need to survive. And the one
Mm-hmm.
way to survive is let's produce some seeds.
Mm-hmm.
And so we didn't carry those seeds really far. We took them down one generation from the parents, and we did not see any change in genetic diversity because that was the one concern is you're going
Yeah.
to start inbreeding your population. But I think if you continue doing the inbreeding, you would start seeing reduction in heterozygosity, but we did not see it after one generation. So seeds that came out of those plants were still pretty diverse.
Mm-hmm. Yeah, you should go back and do some more work on that
Oh,
because I find it's fascinating.
and so did so many people. So
Yeah, apparently, right?
um, that is totally out of my field. That line of research is nothing to do with pathogens, but I had a good graduate student that had a background in genetics, and so
But
she
Zamir, we're all waiting with bated breath for your second paper on
Well,
herming.
now that I talked to you, I'm going to make it a priority.
Oh, good.
I'm going to go back and see if I can at least narrow down what the options might be and what causes it. That's the key.
I'm
I'll do it if it means that growers can avoid stressing their plants and getting male flowers. Let's find out what's causing this and how do we get around it. And that's where I see practical outcomes.
I'm very much the same way. Like applied sciences, answering questions, real questions that people are asking, not
Yeah.
just doing science for science sake. Like I could,
Yeah, there's
yeah.
there's value in that too, but I think knowing you're doing it for a purpose, in my case, get the cannabis industry up and running and produce a better quality product is what I'm after.
Yeah, it's
It,
not it,
no,
it
it's too
for
basic.
yeah, for the real world, for the people who are boots on the ground, that's not really information that's going to help them any. I mean,
I,
yeah.
I mean, there's room for both types of people, but a lot of my funding comes because it's practically oriented, right? They want to know
Mhm.
what the outcome's going to be in a couple of years or sooner.
That's where I come from too, is answering the questions that people need answers to. Because I say that, you know, we're not doing this for fun, although it is totally fun doing science. We're
It's
not
fun.
doing this for fun. We're doing this for you at the end of the day, you know?
And I mean, going back and looking at some of the other work I've done on ginseng and blueberries and so on, the people that I've met in the cannabis industry are absolutely amazing.
Hmm.
Their personalities, their strengths, their attitudes, it's very positive. They really believe in what they're doing and why they're doing it. And not to say that the other growers aren't, but I found I've met more interesting people in the last 7 years since cannabis was legalized than through my entire career. And that goes for people like you and others that reach out to me and ask me things. It's been a really good experience.
I think the whole culture is just steeped in community and richness and storytelling and ingenuity. They've been working with a plant that's been illegal for 100 years,
Yeah.
and they've managed to keep this underground thing going and making new strains and
Oh
keeping
yeah.
legends alive and without much record keeping, which is incredible because you can't
Yeah,
leave a
no,
paper
no,
trail,
it's a fascinating industry.
and stoners are they're so incredibly creative. Like
Yeah,
they just are great thinkers and they
yeah, and
problem solvers,
yeah, oh yeah.
troubleshooters.
Not a passion. Like I said, in the last 7 years, it's been a really good positive experience for me to have
And I
worked with
I've
it.
run across people all the time in the cannabis industry and they say, "Well, I'm not a scientist." And I'm like, "But you are." Like you make an observation, you have an idea of why this is like it is. You test it, you repeat that test to make sure, you
Yeah.
may or may not write it down, but a lot of them do. Or they keep records up here, like I did this and this happened, and so I did it again and the same thing
Oh,
happened.
yeah.
I'm like, that you're a scientist.
Yeah, I know I've gained a lot of information from talking to growers. I mean, they know a lot of stuff and they'll tell you stuff that you can then back up by doing research, right? They've
Mm-hmm.
made the observations, like they've shown me hermaphroditic plants, and then I can take that information and start researching to find out what happened. Or they can give me a plant that's got weird looking flowers that I think is a virus, and I can take it back and try to sort out what it could be. That's
Yeah.
why the interaction is so useful.
This term bro science gets thrown around a lot. And I think that as scientists, if there's somebody who has seen something, observed something over and over again, and they have a theory, but they don't have this, the classical science training to figure out how to figure out what exactly is going on. I feel like as scientists, it's our kind of duty to take this information that they coming to the table with and work on the problem and figure out why we're seeing what they're seeing and use our scientific backgrounds to help answer those questions.
Yeah, I totally
And
agree. Yeah,
yeah,
and this kind of forum is one way to convey the results of what we're doing, right? Is through podcasts and
hmm
seminars and so on. And I like to give seminars as well. Not quite as much as I like to write, but I don't mind getting up there in front of a group of people and telling them what we found.
yeah. Is it too peoply?
Yeah, it's just the stress level is higher when I stand up in front of a group of 200 people than I do when I sit at my computer and I know I'm going to write for 10 hours. It's natural, they say, when you're public speaking or facing people, but then I get over it really quickly, so it's not a problem. But for me, writing is completely relaxing.
That's nice. I used to be terrified to give presentations. Like I look back on some of the presentations that I was forced to give, and I was absolutely terrified. You could hear my voice cracking and like my knees were knocking. Every year we had research day where all of the students in the biology department, the grad students, would present their research. Most terrifying day of my life every year. But when I got into cannabis, I'm like, Anna, you're getting into a research space that people are gonna want to talk to you. They're gonna want to listen to you. You need to get some big girl panties and put them on and suck it up. Figure it out. But I'm much better now at giving
Yeah,
presentations, and I
yeah,
see it more because I used to think like people were gonna come at me and like, stump the scientist, like laugh and point and be like, "You're wrong. You don't know what you're talking about." Which sometimes they do that. That's just rude. But for the most part, it's more of like telling a story, you
yeah.
know?
You got to make it interesting. You got to reach out and convey that information, and it's very important. I mean, you teach, I teach. I find
Mhm.
teaching to be a super important task. We got to do a good job at it, and that's the only way you'll convey information to future scientists.
And especially after COVID and with everything that's going on in the world, there's a lot of trust that's being lost in science. People are just distrusting and think that scientists are all paid off by big pharma and we're all getting these fat paychecks to give them the information that
They, they don't.
I'm
They
not
don't.
rich. Are you rich? I'm not rich.
No, they don't realize the workload that's involved. If you figure out per hour, we don't make a lot, but it's the passion, it's the interest, and for me, it's also the independence. To be able to get up 7 years ago and say, "Oh, I'm going to be working on cannabis now that it's legal," and drop my work on blueberries, whatever. I'm not stuck to one area of research that others might be. And so it's worked really well for me to be able to explore. I don't think there's anything after cannabis. I can't think of another plant that would come along and make me abandon my work in cannabis because it's just so fascinating.
It is. It really is one of the most fascinating plants in the world. And I say, arguably, the most recognizable plant in the world. Like,
Yeah,
everybody knows what a
yeah.
sunflower is, but you put a cannabis leaf in front of most people, they're gonna be like, oh, that's marijuana, that's ganja, that's, cannabis. Almost everybody in the world can recognize a cannabis leaf.
And what I'm finding too from some of the emails I get is that other countries like Thailand and Brazil and Germany and others that are exploring legalization are starting to look to North America for their advice and for their input because we've been at it, a little bit longer. And
Hmm.
that's nice to see as well that the word is spreading to other countries that potentially are facing same disease problems. They're going to get hermaphrodite plants where they are. Maybe they'll see salt crystals on their plant. The word's going to get out to other parts of the world.
And well, yeah, I hope we can give them good information. I feel like, at least in the United States, it's kind of a mess still.
Yeah.
But, I think that the more people that start working on it, it'll start getting better, or, start figuring out some of these
Yeah.
problems. And
Well, working
um,
and sharing. I think that's important to share.
yeah, I agree. Collaborations and sharing of information so that we can all just work it, yeah, work together on these things. Is there anything else that you want to discuss or talk about or ask me?
I mean, we've covered a lot of ground. We've gone through diseases, tissue culture, hermaphrodites, hairy trichomes, toxins. I think we've done really well. I think you've
Yeah.
covered most of the projects that I have ongoing. The one that I'm working on now that I guess you'll see eventually is this crown gall disease. Agrobacterium causes crown gall on cannabis, and we've just found that last year. And that creates some really interesting phenotypes on the plant. There are plants that are a little bit taller because there's hormones produced by the bacterium that cause weird things to happen. But the most interesting is I was seeing more male flowers again on these stressed plants that were infected with this bacterium. So I may be bringing in the hermaphrodite story back in, but now triggered specifically by a pathogen that is infecting the plant and producing these hormones. So again, it goes back to this hormonal idea of
Mhm.
what may be causing plants to produce these banana things. But so it ties in well together. So
Yeah.
the story continues in terms of what else we may be looking at.
All right. Well, I'll be counting your papers as they get published
Well,
this
thank
year.
you.
I've got a notch on my bedpost for every paper that Zamir puts out. And I was tossing around the idea of going to CanMed, but I don't think I'm gonna go. You
Oh,
usually go there.
I'm
Uh,
going. I want to go to California. I haven't been for a couple of years. And Lake Tahoe is a beautiful area. So I didn't go to last year Puerto Rico. I just couldn't make myself travel to Puerto Rico. But California is fairly close to me. Just
mhm.
flying to Sacramento, it's 2 hour flight, so I will be going.
Yeah, I hear it's a great time, and I want to go because I have a bunch of friends that are going. And I think, I mean, you're well known, you're on the circuit. I'm sure that you're gonna meet up with a bunch of people that you know. It's so nice to see. I'm a weirdo. I think of conferences now as like I get to see all my friends instead of like work.
Yeah,
Right.
I'll be going. So, and I'll be presenting the salt trichome thing that you just mentioned at this conference. Yeah.
That's so cool. I mean, I know about guttation and it
Yeah,
pushing out water and,
I
and
like,
sugars,
thank you. That's the word I was looking for. Guttation is where the plant forces out sugars and other things. This is exactly the same thing. You
but with
see the
salt.
bud with salt. Yeah. Too much
Yes,
salt. Too much salt.
super interesting. I didn't know it could do that with salt. All right. Well, if you have nothing else, hey, you are so awesome and you're always very generous with your information and sharing, and I've asked you for photos of things before which you've generously given to me, but I know there's people out there that might have questions about your research or anything that you've written, or just, how do they sign up for your class in Canada? I don't know. Where can people find you? Because you're not super active on LinkedIn. So
I've got a website. Yeah. So if you go to Simon Fraser University /biology/Punja, you'll be able to see pretty much all the works that I have and various other things, links to articles and so on. I was on LinkedIn for a while and then somebody hacked into my account and I don't know what they ended up doing to it. So I had to restart all my connections and contacts and so on. So I don't keep up with that like I probably should because I met quite a few interesting people when my account was up and running. So
mhm.
maybe I'll go back to it. Maybe that's one thing I can take away in addition to everything else we've talked about is reactivate my LinkedIn account so people can get hold of me.
Yeah, there's quite a good network of cannabis
There,
industry, cultivation, and science folks on LinkedIn that
there is.
share really good information, new papers that come out.
Yeah.
So yeah, all right. Well, I'll put your website up on the screen so people can find you. And
Sure.
then Google Scholar is another really great place to find your publications. So just search for Zamir Punja, an author, and then it'll list all your
Yeah.
publications. You can sort them by popularity, you can sort them by year published. That's where I looked at your extensive publication list, and I had to kind of pick out which ones I thought would be interesting to talk about. But
Well, I appreciate.
congratulations on all of your research, and it's awesome. I think you get to do such interesting stuff.
Yeah,
Um,
and I appreciate the opportunity to talk to people about it, especially you.
Thank you so much. It has been a wonderful conversation. Thank you so much for your time and
Thank
words
you.
of wisdom. Loved it. And I can't wait to talk to you again sometime.
Sounds good to me. Thanks a lot,
All
Anna.
right. Thanks,
Be
Samir.
safe.
Have a good day. Bye.
Bye-bye.